So thank you very much, Heiko, for this kind introduction, and welcome back from this delicious lunch to my talk now about automated expansion cryopreservation at scale. First of all, I would like to give you a short overview. So in a first point, I would like to define the needs and requirements, what is necessary for using iPS cell technologies for next generation drug discovery applications. Then I would like to give you some suggestions about technical solutions for future applications. And here I would like to focus mainly on the iPS cell expansion and also on the biobanking. And finally, as outlook, I would like to give you a hint about the topic large-scale differentiation, what is possible here already, and new cell model generation using new and innovative materials. So we have already heard today a lot about the possibilities and advantages of iPS cell technologies. So until now, it's the biggest advantage is that you can generate iPS cells with a patient or disease-specific background, and then you can differentiate the cells again into the cell type you would like to use and you need. And then these cell models can be used in terms of disease modeling, candidate screening and drug development, and also toxicity tests and efficacy. But really the point is to use iPS, this very good cell model of iPS cells in new drug development applications. The question is, what is still missing and what has to be realized to make it really a success story? So the point is, what you need for future applications is you need at first high cell numbers to really make your screenings and to test your drug candidates. You need a high rate of standardization and reproducibility. The documentation has to be very good during the whole process. You need a scalability without compromise in biological quality. That means it should not have an effect if you cultivate your cells in a small petri dish or even in large T175 flasks. Of course, your workflow and the cell production and the cell workflow has to be robust. You should have a high flexibility and also transferability of the workflows you're using and about the technologies you're using. And you need a stable banking and logistic with seamless cold chain to really guarantee the quality of your cells for future applications and also not only at the time when you need them, but also further on. And finally, you need a positive economies of scale. That means if you're doing the necessary upscaling of your process, then there should be in parallel a reduction of the costs that you have here. So what production technology of cells could meet all of these specifications? Until now, the production technology that is used is, you know how it looks like. It's a normal clean bench, and you have a technical assistant who's doing all the cell culture. The big advantage is usually you have small cell culture plates or dishes like these here. With them, you have a high growth area volume, so you have a good nutrient supply, and during the whole process, a minimal physical stimulus like shear stress. Because here I would like to state that especially iPS cells are very sensitive to shear stress and really react directly on it. And with this standard procedure and standard workflow, you have a monolayer culture in 2D, like you can see it here. With this monolayer culture, you have short transport and diffusion paths. You have a very good gas supply and nutrient supply, and you have hardly any gradients. If you now want to make a scale-up of this process or to reach the high cell number you need, the only possibility until now is to make something like this. You have to duplicate your technical assistance, and we all know that this is really not the solution at all, because finally, especially in the iPS cell cultivation, you need some kind of golden hands. So some technical assistance is getting better cell cultures than other ones. So the point is the only possibility to really meet the demands of a future iPS cell technology application is you need automation solutions, especially if you're having a look on these economies of scale. If you're only duplicating your staff, then the costs are also duplicating. So automation is the best solution here. Having now a look on this example for iPS-based cardiovascular drug development, you see here these are in the normal working steps. You have a patient recruitment, somatic cell isolation, reprogramming, differentiation, and then your drug testing. And now the question is where everywhere in this workflow you could involve automation. So you can involve automation in the terms of reprogramming and clone picking, for expansion of cells and differentiation, and also for the banking of cells. Within EBiSC, I did some kind of prioritization of these different possibilities that you have, because especially for EBiSC, the main priority was on the expansion of cells, on the banking, and also later on also on the differentiation of cells in an automated way. Therefore, I would like to focus on the next slides mainly on these three topics here. If you're now talking about automation, what automation solutions do we have at all? We have the possibility to use bioreactors for the expansion of cells. Here the advantages are that we have an improvement of a very good volume to growth area ratio, that usually the expansion rates are higher than in 2D. But the disadvantages here are that for bioreactors, you really have to make a completely new protocol development, because your standard SOPs that you have for your manual culture cannot be transferred in a bioreactor. That's not possible at all. That means you need some time to develop and adapt your new protocols. And also standardization is quite difficult, working in this large volume. Another possibility is that you use robotic platforms. Here the advantage is that you, in most of the cases with some adaptations, you can use your standard SOPs as base for your further developments. That means direct transfer of the protocols is possible, and you have a complete process monitoring and standardization. The disadvantage is here that you have quite high costs, and that it's quite material consuming, these robotic platforms. They are very cost intensive. Most of these industrial robotic platforms are in the range of 1 million, something like that. And until now, they are also very error prone. That means it takes a long time until you have a stable protocol running on these systems. And then you have only a low flexibility. If you want to change something, then you should think twice about it, because then you start from the beginning. So if it's running, then don't touch it again. And third, especially for the biobanking, you have the possibility to use innovative biobanking technology. With this, you have the advantage that you have also a complete process monitoring standardization. These new systems guarantee a closed cold chain, and you avoid sample mismatches. Disadvantages here: most of these new biobanking systems have, in some points, a lower storage capacity, and they are also quite cost intensive. In parallel to this, in all of these different technologies, you have to make a protocol optimization. That means that you have to adapt your protocol in terms of what coating you're using, cell passaging protocols, if you're using single cell dissociation or not. That means that these technical solutions always need some biological protocol optimization in parallel. Having now a look on some examples for full workflow automation using industrial robotic platforms that we have evaluated within EBiSC. You have different categories that can be used. You have some kind of complete liquid handling platforms. Here, for example, you can see the TCAN Evo station. Here you have separated the liquid handling. You can see here the pipettes, then that's doing all the microfluidic stuff. And within the system, you have separated a robotic arm that's doing the labware positioning or the positioning of your cell suspensions. These systems are often quite flexible. That means you can use all formats, all plate formats and flasks within the systems. Another possibility is to use pick-and-place robots. Here, for example, the tab now Sartorius Compact Select. And here the innovation is that all the human working steps are imitated. That means you have here a robotic arm, and this robotic arm is later on taken a pipette here and here, and making the liquid handling like a human arm would do the same movement. These systems are not so flexible in the format. Usually you only can use T75 and T175 flasks, so they are not combinable with cell culture plates. Both systems can be used and are already used for iPS cell expansion and also for differentiation protocols. And finally, there are several modular platforms available, like here the stem cell factory that was also part of within EBiSC. And here the point is that different off-the-shelf systems are combined and connected. So, for example, you have a pipetting station, then you have a microscopic station, or even clone picking was possible here, integrating a system called cell selector. So with this system, you could had a possibility to even make a reprogramming, iPS clone picking, and expansion. But the disadvantage of all this system is that you have really high initial and also maintenance costs, and you need a permanent internal support of engineers, because as these systems are quite error-prone, you always need some engineers beside the system to keep it running or to help if there are some errors within the system. So beside now these robotic platforms, of course, you can also use bioreactors. There are different categories of cell culture bioreactors. Of course, static, also cell culture flasks are a quite simple bioreactor, or you use dynamic bioreactors. Especially for pluripotent stem cells, these diet bioreactors are most commonly used. And here, mainly the spinner flasks. Within these stirred bioreactors, most of the people use aggregate formations for the expansion. That means you seed in your single cell suspension and make then a bioreactor protocol, and then you expand your cells as aggregates. What you have to take a look on is that you need to control the aggregate size, because otherwise you have a spontaneous differentiation. And sometimes it's also quite difficult to make a dissociation of these aggregates at the end, because you need to make a single cell passaging to start the next bioreactor process. And if these clumps, the aggregates, are too large, then it's always quite difficult to dissociate them again, finally. Nevertheless, here also within our consortium, there was already quite success to establish iPS expansion using aggregates in suspension. But nevertheless, using these kind of protocols, you are losing the advantages of your standard, established, very good working 2D monolayer culture. That is using in a normal cell culture, using petri dishes, flasks, etc. Having only a monolayer with one cell layer, with short transporter diffusion paths, and guaranteeing a very good nutrient supply of the cells. Therefore, our idea was now, if it's possible, to bring a petri dish or a cell culture flask in the bioreactor. How could this be realized? So our strategy was here to first of all have a look on the surface chemistry of the treated standard cell culture flask. You see it looks like here you have some hydroxyl groups, some carboxyl groups, phenol groups here. And then to bring these surface chemistry on the microcarrier to give now the cells the possibility to grow as 2D monolayer in a bioreactor. Of course, we could have started with normal plastic microcarrier or a polystyrene microcarrier, but to be directly from the beginning as close as possible on these active groups here, we decided to use a hydrogel called alginate from marine seaweed. And these alginates, you can see here how it looks like, and we also have here already most of the functional groups that presented here in the surface chemistry of the treated standard culture flasks. The only thing that's still missing are these phenol groups. So we developed a strategy and procedure to couple a linker with phenol groups to the alginate microcarrier. And with this technology, it's now able to use this alginate microcarrier, that the alginate microcarrier have the same surface chemistry than a cell culture dish. And then you can bring your adhesion proteins just with adsorbents on the microcarrier. And with this, you are very flexible. You can use matrixyl, laminin, vatonectin, everything that you can use in your standard, with your standard protocols on your standard flasks and plates is now possible with this technology. So now this is the way to transfer the established 2D monolayer culture in the bioreactor system. And to make it as a proof of principle or proof of concept, we decided, okay, now we have this microcarrier system, and we want now to transfer the EBiSC banking procedure into the suspension bioreactor. Therefore, this is the official EBiSC banking procedure, how it looks like. So usually you start with a vial of one frozen iPS cell line. This vial is thawed. Then first of all, you bring it in one cell culture plate of a 6-fold plate. You expand it for 3 to 5 days, something like that. And then you passage it and split it to 4 wells. Then another passage into 2 75 flasks. And then another passage 4 flasks, harvest, and then you bank 50 vials. And usually you have between 1 and 2 million cells inside of your vials afterwards. So now we have the microcarrier we want to use, we have the workflow, and now we had to decide on the bioreactor technology. Therefore, as I already mentioned, we have this biopolymer technology at Fraunhofer IBMT. We use a quite special alginate where we have the complete production process. It's fully characterized from the harvest of the algae until really the production of the final alginate. From this alginate, we produce beads or microcarriers. Then they are biofunctionalized. Of course, we can also use the alginate as membrane, but here we use them as microcarrier. And currently, we also work on making these alginate microcarrier stimuli responsivity. And now, as automation technology, we decided for the biolevitator automation platform from the company OLS. You can see we have in total 12 of these systems at IBMT. Here you can see on this picture, you can see 6-1. And these inside there, you have 4 of these cultivation chambers. They are looking like 50 ml Falcon tubes. The big advantage here is that this system is not working with impellers like normal suspension bioreactors, but they have baffle inside. So the shear stress in this system is very, very low, and that's why it's working quite nicely with the iPS cells. And with our 12 systems, you have 4 cultivation units. That means we have all in all 48 independent cultivation units. So this is now how the transfer of the biobanking, of the banking procedure looks like. We started with 1 vial of frozen iPS cells. We added here 25 square centimeters of alginate microcarrier in one of the slivet tubes. These cells have been cultivated with a special protocol, of course, in a biolevitator for 3 to 7 days. We did 1 passage and then splitted the cells on alginate microcarrier with now a cultivation area of 140 square centimeters. Cultivated it again for 3 to 7 days, and then we harvested the cells, and the aim was that we really receive 50 vials and can make the banking of the cells. This workflow now was validated with 3 different cell lines and triplicates. We used these 3 cell lines here, and what we can show is that it was possible with using this protocol to produce 50 vials within 13 days. So you can see here even 11 days, here 13 days. and that we also receive the cell numbers that we need. So at the end, it was possible to produce 50 vials with 1 to 1.5 million cells per vial. So we could fully fulfill the EBiSC specification using this setup and this protocol. Of course, we have taken a look on the stemness finally, even after cryopreservation and thawing again, and you can see the stemness is the surface markers, the stemness markers are still there in comparison to 2D. We also did some qPCR, and these approved the results you can see here. So here we can state that using this automation workflow, it is possible to transfer the EBiSC workflow into the suspension bioreactor. Of course, the biological results are quite nice, but for EBiSC, it's also important to have a look on the productivity and the costs. With our system, it was possible, if we are talking about passaging and expansion, for the complete banking workflow, it was necessary to only make one passaging, and then we were able to receive the necessary cell lines latest after 15 days, so usually after 13 days, but latest after 15 days. Doing the complete procedure manually, you have 3 passaging steps, and you even need much longer time. That means in total, we could show that we had a time reduction of 25%, and also that the personal costs are lower because we only had to make one passaging step, and we also only have one cell culture bottle. So we only had one levy tube, and doing it manually, you have several flasks, dishes, and so on. Having a look on the medium consumption and the matrix consumption, you can see the same picture. As we started at the beginning with a very low volume for the inoculation of the cells, we could reduce our medium consumption to 70%, and also the matrix consumption could be reduced significantly due to the point that we only had one passaging step, and of course then you also need less protein coating and protein adhesion proteins. Okay, now, until now we had a look here mainly on this point, that it was able to make here the automation of this workflow. And now I would like to have also a look on the banking procedure here in the end. The mirror bank at Fraunhofer IBMT, we are using improved cryotechnology, the so-called protected food system. The big advantage here is that you have systems looking like this. You have here in the lower part a standard liquid nitrogen tank, but here above you have this so-called protected food system. This protected food system can be cooled down to -100 to -130 degrees. That means if you bring your samples in or out, the samples are always cooled, and there are no only low temperature changes, and the cool chain is not interrupted, meaning that that all samples here in the tank have a stable storage environment. With this, we have been able to guarantee the complete banking process with a closed cool chain. That means, as mirror bank, we usually receive our samples from the central facility in a dry shipper. Then we have another technology, the so-called cryo workbench. This cryo workbench it's the same principle. You can here cool down the complete workbench to -100 to -130 degrees. That means we receive the samples in a dry shipper, bring them directly here into this cryo workbench. That means the samples are still cooled, and in this cryo workbench we can do the sorting. We can also, even in this cryo workbench, we can register the cells in our database and integrate it. And then we have an automated storage system, so the samples are then here bringed in a cooled transport container inside the cryo workbench and are going then into our mirror bank. Meaning, as I said, that the cool chain is not interrupted during the complete shipping process and storage process. To show you what effect could be if you have not these seamless coolant chain, we did some experiments with iPS cells. These are first preliminary tests, but the idea was to changed the temperature of the cells during storage. That means we brought the cells 400 times from minus 130 degree to minus 60 degree, and to see what happens with the cells. And the point is, you already can see here, we had no look on the viability, because usually viability is always quite high after cryopreservation. We had taken a look on the adhesion rate, because only when the cells adhere, you can see if they are functional anymore. And what you can see without cycling, we had here adhesion rate about 40 to 50%. But after this 400 cycles, we only had an adhesion rate of about 20%. So these tests should show you that it's really important to have a very good quality of the samples, that you have a look on the storage and of the temperature profile during your banking system. Here's a summary of the mirror bank performance. Until now, we have 190 iPS lines in the mirror bank. All EBiSC SOPs are established. Also for the QC, we have everything in place at Fraunhofer at the mirror bank. The complete system is running under a quality management system. It's the ISO system. And we have a LIMS integration that's still in progress, but that will be done until end of the project. Our capacity is that we have we have a fully automated high throughput cryobank with 3 automated transports. We can handle 144 vials in parallel. That means we can bring them in, bring them out, all completely automated. And we can store 60,000 vials, but this is can be scaled to 600,000 vials. So now, as I'm running out of time, I would like to give you only an outlook about the work that's still in progress. So we are also dealing with the differentiation of cells using automation procedures, and this will be mainly done from next year on in the Fraunhofer Project Center for Stem Cell Processing Technology in Würzburg. We have received here a Fraunhofer project in the range of 20 million euros, and the aim is to develop new processing technologies for stem cells in combination with innovative materials. And for example, we are doing differentiation also in a bioreactor in NPCs, so in the neuronal direction of the cells, but also into cardiomyocytes. And the point is that with the biolevitator, we have the possibility to make a differentiation of 50 million cells at once. So the upscaling is quite nice. Comparing to the hanging drop, you can also see that in the biolevitator, connexin is much more expressed than in the standard hanging drop. So it's possible to also make a large-scale differentiation in the bioreactor. And this last slide, I would like to show you the possibility to use new materials or the impact of new materials on your cell workflow. So here the point was to make a better cardiomyocyte maturation using new materials. What you can see here is, again, an alginate disc. On the alginate disc, cardiomyocytes derived from iPS have been seeded. And this here is now in culture without any passaging for over 100 days. I think now it's 120 days. And you still have this very nice beating, and it's they're beating very in parallel. We have a better cellular organization and sarcoma maturation using these alginate scaffold, and also a higher functional recruitment. So with this outlook, I would like to thank you and open for questions.
Thank you very much, Julia. We are limited in time, so maybe only 1 or 2 questions. Are there any questions? There's a question.
How these iPS cells, do you remove them from these beads?
Yeah, we remove it from the beads.
And what's the efficiency of that?
Yeah, the point is that one of the advantages of the alginate is that there is one enzyme that's really only diluting the alginate itself. That means with this, we can really dissociate the alginate microcarrier and have left over the iPS cells without using enzymes like trypsin or something like that, that's detaching the cell-cell contacts. So that is one possibility, but also ETTA, then the standard methods is possible for detaching the cells, and it's the efficiency here is quite high.
Maybe one more question, if necessary. Otherwise, there's a coffee break for discussion. So if not, thank you very much, Julia, again.